Okay, good evening, friends. So this is the fifth class. Tomorrow will be the last class as far as this plant disease management is concerned. So in yesterday's class, we discussed or we learned about what we call various measures to control pathogens and pests. So whenever I say pathogens, it means disease causing organism could be a fungus, a bacterium, a mycoplasma. And pests means mostly insects of various kinds, leafhoppers, aphids, thrips, jossettes, their caterpillars, their adult ones, etc. They could be, sapsuckers or stem borers or, fruit borers, etc., etc. So primarily, this management, that is, this pest management involves two things. As you know, one is a preventive measure, the other is the curative measure. So preventive measure means here we take all precautions. All our precautions are concentrated avoiding the entry of the pathogen into our area. That means into our field. We see that we check the entry of the disease causing organism into our environment, that is into our field. So in spite of taking all measures, because the pathogens are very small, too small. So the fungal spores are too small. The bacteria are very small. The viruses are very small. The mycoplasma are very small. So it is very difficult for us to check their entry into our environment, that is into our crop environments. So in spite of taking all measures, if your crop is affected means, then you go for what we call a curative measure. So curative measure means here, when the onslaught, when the pathogen enters, when the pest attacks your field, then we have to take measures to control their population, to keep the population of disease-causing organisms below the threshold level. See, our aim is not to completely eradicate them, not to eliminate them. So our aim is not elimination. Our aim is to check their population that they remain below threshold level, means the loss caused by them to the farmer or to our field is minimal. So why? Because elimination we don't go for, because if we have no right to eliminate any organism, because we are not the creators. So only creator has the right to eliminate. So our aim is to protect ourselves, to keep ourselves aloof from the onslaught of the pathogen. So in that, we discussed, in yesterday's class, we discussed about various principles of disease management. In that, I told you the meanings of various important 6 terms are there. One is avoidance, the other is exclusion, the eradication, protection, immunization, and therapy. So avoidance tells you avoid. We take all measures that the pathogen does not reach our area, our environment. So for that, generally, we go for what we call various agronomic practices. So second one is exclusion, means we check the entry by following certain legal procedures, certain quarantine procedures, certain inspection and seed certification and seed treatment procedures, so that the pathogen does not make an entry into our field. Then third one is eradication, means here if you tried for avoidance and exclusion, still the pathogen makes an entry, means then you do eradication. In eradication, generally we follow various agronomic practices such as crop rotation, sanitation, roguing, soil treatment, fumigation, heat, chemical, etc. We use various measures to control the spread of the disease. Then the other measure is protection. Your protection is We will read that today. Protection means by using various chemicals. By using various chemicals, we try to control we try to kill the pathogen. We try to check their reproduction. We try to check their spreading, so that the disease remains, or the pathogen population remains below the threshold level. Then immunization and therapy is there. Immunization, of course, we will read about that in tomorrow's class. That is, it is related to your genetics. That is, recombinant DNA technology, or DNA technology, or genetically modified, or hybrid varieties, disease resistant varieties, etc. We discuss about that in tomorrow's last class. So then final one is, of course, therapy, wherein we use various chemicals or biological organisms to control the spread of the disease. Okay, so in yesterday's class, apart from these principles, we discussed about what we call the cultural methods, the mechanical methods, and physical methods. We discussed it. So cultural means various agricultural practices, various agronomic practices we follow to check the spread of the diseases. In that, we discuss about the ploughing, importance of ploughing, importance of tillage practices. We also give importance to the time of planting and keeping your field clean, that is sanitation, then going for crop rotation, then thinning and pruning. Thinning means maintaining the distance between one plant and the plant, or between the rows. So, and what we call that is thinning. Pruning is cutting, separating the diseased part or the fallen parts, or your leftovers of the previous crop, etc. You burn them, you bury them. That is, pruning. Then we also learnt about, judicious use of fertilizers. So fertilizers, especially nitrogen fertilizers, are to be used very judiciously. They should not be given too much nitrogen for the crop. So it has been, proved. Research has proved that if you give extra nitrogen, you are attracting more pests, so more damage. So hence the nitrogenous fertilizers are to be given judiciously and in split doses. And we also discussed about water management. So here, water management in two forms, either alternate flooding and alternate drainage. These two practices, especially certain soil-borne, nematodes, etc., are allowed to come out and also get killed. Then we also discussed about various hosts, various alternate hosts such as Cyanodon or Citeria species and Panicum species, etc. They act as alternate hosts for various pathogens, various rusts and smuts. They are to be removed. Then we discussed about mechanical. These are all cultural or agronomic practices. Second method was, mechanical method, wherein you physically uproot the unwanted plants, or you physically break the diseased parts mechanically using, power, mechanical. And also you erect various traps. The traps could be, fly traps or sticky traps or repellent traps, etc., or even laying of mulches, especially polythene mulches, etc. By doing all these things, you will be checking the spread of the disease. So this was regarding mechanical method. And the third method we learned about physical method. So physical methods means primarily use of environment. So air, water, and sunlight. These three can be used to check the spread of the disease or to keep the pathogen load below the threshold level. So here, application of the heat, hot water can be used, hot air blowers can be used, and soil solarization. Soil should be exposed to hot sun and deep ploughing, etc. can be done. And exposing the harvested seeds or diseased seeds or fungal infected seeds, they are to be exposed to hot sun. Seed treatment has to be done. Seeds are to be properly dried before sacking, before storing, moving them to the storage or godowns. They are to be properly dried under hot sun so that the moisture content will be at minimal level. So if the moisture content is at minimal level, they can be stored for longer duration. So these three techniques we discussed. One is agronomic, the other was mechanical, the third one was what we call physical methods. So now in today's class, we discuss about the methods of, that is curative methods, especially curative methods means here, instead of taking all these precautions, the disease causing pathogen or pest has taken entry into your field. Then we should go for curative methods. So preventive methods and curative methods. So preventive methods we discussed, then we move to curative methods. So curative methods, generally, primarily, there are two types. One is curing, that is checking them by using what we call chemicals. And the latest one, environmental friendly method is what we call biological method. So first we learn about a chemical method. See, I told you in my first class itself, the chemical method should be used as a last resort. Chemical should be used as a last resort to check the disease or the pest menace. Why? Because excessive use of chemicals not only increase the input cost for the farmer, but their damage to the environment is enormous. They are polluting your environment. They are polluting all your foodstuffs. So hence, they're causing irreparable loss to our environment. The air. They're causing air pollution. They're causing water pollution. They're causing your water, soil pollution. And even the remnants of chemicals are found in the foodstuffs that we take. All types. It could be cereals, it could be pulses, even it could be what we call your milk and milk products. The fish, the mutton and chicken, whatever you eat, even when the food gets contaminated, that enters into the animal body. When you take that animal food, it enters your body. It gets deposited in your adipose tissue. That leads to what we call biological magnification. So your soil is getting polluted. Air is getting, water is getting polluted. Your foodstuffs are getting polluted. They are getting polluted to irreparable doses. Hence, one should go for this chemical method as a last resort. Okay, so now in today's class, we confine ourselves to the chemical methods of pest control and disease control, and also biological methods of pest and disease management. So first of all, we will learn about chemical methods. Many chemicals. So how do the chemicals help in controlling this, pests and diseases? So they help, they attract, they reduce, and they destroy the disease causing pathogens and also the pests. So generally, they are called what we call biocides. They kill the living organisms. See, because it could be a bacterium, a virus, it could be a what we call a mycoplasma, it could be an insect. So it acts against them. The chemicals act against them. So the history of chemical control. The history of chemical control back to 1800s. That was developed, that was produced was in France. It was Milordet. Milordet was the man who first Bordeaux mixture. Bordeaux mixture was produced in 1800s in France by Milordet. The Bordeaux mixture is nothing but copper sulfate and calcium hydroxide mixture. It was found to be quite effective in controlling downy mildews. Later on, it was used on various, even fungal and other pests. It was able to control the spread of the disease. So later on, in these 100 and 150 years, thousands of pesticides, insecticides have been developed. So especially the organic pesticides, n number of organic pesticides are available in the market now. Both organic and inorganic pesticides are available. Insecticides and pesticides are available. So based on the target organism, the pesticides are, classified. For example, fungicides. The chemical that check the fungal disease is called fungicide. Insecticide that checks the population of insecticides. Herbicide that checks herbs, that is weeds. Weedicide or herbicide. Then bactericides. Bacterial diseases are checked. Bacterial population is checked. Then nematicides, as the name tells you, nematicides, nematohelminths, roundworms, hookworms. So their population is checked. Then algicides, then what we call molluscicides, mollusks. Then avicides. What do you mean by avicides? Aves means birds. That act against birds are called avicides. Then rodenticides. Rodents means rats. So now, so many chemicals are available to check these rodenticides. So generally, we face the, especially the fungicides and pesticides and rodenticides, not only on standing crops, but also in godowns. Godowns, we make use of all these, chemicals to enhance the, storage of our stored products in the godowns. So now in India, you will be surprised to know that 50% of the pesticides are used as a protectant for your cotton. So 50% of the pesticides used in India for protecting cotton pests. And of course, it is followed by 18% of the pesticides are used to protect the rice crop, then 14% for vegetables, and for plantation crops, 8%, and for oilseed, 7%, sugarcane, 2%, of course, and others, 1%. So by this chart, what we understand, 50% of the insecticides and pesticides or chemical substances are used as a protectant for cotton. Okay, so now these chemicals, have what we call an active principle. Chemicals have an active principle, and that active principle is what we call formulated for, say for example, the active principle that is used to control our body. So if you suffer from what we call temperature, fever, then you go for paracetamol. This is the active principle. But trade name will be different. For example, Crocin or, Vicks Action 500 or Dolo. These are all trade names. So the manufacturer gives his own specific trade name, but the active principle is same. So nowadays, even the government of India, generic medicines. So that is the generic medicine is the active principle, but different companies use that active principle and release the medicine in different names. That name, what we call a trade name. So here, what we have an active principle and what we call and they are released in trade names. And generally, we have what we call these names. These names generally these names are chemical names. Names these are chemical names, and generally the names for these chemicals given by what we call IUPAC, that is American Chemical Society, or what we call International Union of Pure and Applied Chemistry. See, you know, ICBN, International Code of Botanical Nomenclature. This is the body that regulates naming. So also in chemistry also, different chemicals have names, and these names or nomenclature is regulated by an international union. So different companies release their produce on different trade names, but the active principle will be same. Okay, so now we have primarily we have 4 groups of, though I told you we have avicides, we have rodenticides, molluscicides we have, nematicides we have, fungicides we have, insecticides we have. So many are there, but we have only in our syllabus 4 groups of these chemical pesticides are there. One is a fungicide. The fungicide against fungus. Then bactericide against bacteria. Nematicide that acts against nematodes. Then the last one is pesticide against pests. So we have in our syllabus these 4 groups of chemicals. Let us know about them one after the other. First of all, we'll begin with the fungicides. I told you, I think you remember. So most of the diseases on our crop plants are caused by fungi. So fungal diseases outnumber the diseases caused by all other pathogens put together. Hence, we concentrate more on fungicides. Now, in fungicides, we discuss about these fungicides, generally fungicides are classified based on their mode of action, their mode of mobility, and also based on their chemistry. So first of all, based on their action, they are classified as broad spectrum fungicides and narrow spectrum fungicides. Broad spectrum means they control various fungal diseases. It could be a phycomestes, it could be ascomestes, it could be a basidiomestes, rust, mutts, oomycetes, etc. So broad spectrum act against almost all types of fungal diseases. Whereas narrow spectrum means they are quite specific. One will be effective on a rust, or one will be effective on a smut, or one may be effective on a powdery mildew, or the other may be effective on what we call a downy mildew. So if each pathogen group has a specific chemical controlling them, such chemicals we call narrow spectrum. If it has the ability to control various fungal diseases, that we call what we call a broad spectrum fungicide. And based on their mobility, the mobility, some are what we call local and the other is what we call systemic. Local means, so if you're spraying on the leaf means it remains on the leaf and it checks the spread of the disease on the leaves only. Then while systemic means if you spray that particular chemical, it enters into the conducting tissue and it gets, it will be carried to different parts of the plant body. That is what we call a systemic, a contact or a local one, or a systemic. Then based on the chemistry, they're called inorganic chemicals and organic chemicals. So the primarily based on mode of action, mobility, and chemistry, these fungicides are variously classified. See, for example, based on their mode of action, that is broad spectrum and broad spectrum and narrow spectrum, we have what we call mancozeb is there. So mancozeb is one which is what we call, it is effective on a single site, local. And there are, based on mobility, some are, for example, mancozeb and thiram captan. They are what we call local. Whereas systemic bonomil is their carbendism. These are the chemicals that fall under what we call systemic. If you spray carbondism, if you spray what we call bonamil on a leaf, it even transmits to inflorescence, copse, and also even into the root system. Whereas the local one, Mancozeb and Thyram, Captan, they are local. See, generally Thyram and Captan generally we use for as a seed dressing. Seed dressing. So if your seed has fungal spores, if you treat them with Thyram Captan, whatever the fungal spores that are there attached on the test and tegmen of the leaf gets killed, get eliminated. So based on the chemistry, I told you, based on the chemistry, generally they are what we call, um, you know, uh, organic and inorganic. So organic and inorganic are there. This is another way of classifying. And also based on the chemistry, of course, organic, inorganic, and organic, inorganic, a good number of varieties are there. For example, inorganic, or even in inorganic, we have what we call borax mixture, that is copper containing. Then we have, sulfur containing, sulfur dust and sulfur lime. We have, sodium bicarbonate is there, and monopotassium phosphate is there. These are all inorganic, under inorganic compounds. So inorganic, borax mixture is there, Blitox is there, microthiol is there, thiolux is there, and what we call sodium bicarbonate is there, monopotassium phosphate is there, dipotassium phosphate. These are all inorganic, fungicides. Inorganic fungicides. Then organic fungicides. These organic fungicides outnumber inorganic fungicides. Organic fungicides are many. They're more in number. And generally, here, organic fungicides, they generally, they may be based on their organic, they may be quinones, they may be aromatic compounds, they are heterocyclic compounds. And, a good number of organic, compounds are there. And few to mention. See, for example, thyram, an organic, a sulfur containing. A sulfur containing organic one is what we call thyram. And generally it is available in the vegetable powder. And generally it is used as a seed dresser. Then, for example, Mancozeb. This is what ethylene containing carbamates, and generally diethane M45 is there. And this generally used to control fungal diseases such as, leaf spot of, groundnut, that is Tika disease, can be controlled. And even we have Maneb is there. This is used to control various fungal diseases appearing on tomato, potato, and vines and flowers, trees, etc. Then aromatic compounds such as pentachloronitrobenzene. See, these are all, you have to by heart certain names. So classifying them: inorganic, under organic, under inorganic, various ones, and under organic, various compounds. So like that, you have to classify them, and you have to learn at least 2 or 3 under each category. So, for example, heterocyclic compounds are there. For example, captan is there, and redomil is there, organic, and benzomyl carbendism is there. Another one, generally, this is used, this is a broad spectrum fungicide, and generally soil-borne diseases and also powdery mildews, it checks the spread of the powdery mildews. Then phosphate containing ones. So phosphoric acid is there, then organophosphatic fungicide, generally powdery mildews and leaf spots of rust and smuts, you can check them. So, and so these are some of the, what we call the fungicides. The fungicides, and which are, they are used, mostly in 3 forms. Generally, they can be used in the form of powders, They can be used as emulsifiers. They can be used even for treating your soil, foliar spray, etc. are the forms in which these various fungicides can be used. So then, second group, what do we call antibiotics? Antibiotics are used to check the bacterial population. So we know bacteria cause a number of diseases. We have seen Blastophilis, Xanthomonas oryzae. We have seen Xanthomonas axinopodis citri that causes citrus canker in lime, sweet lime. So these are Erwinia keratovora, soft rot of carrot and others. So these fungal disease, these bacterial diseases can be controlled by using various chemicals, and the chemicals that are used to control bacterial diseases called bactericides. So we have a number of bactericides, and generally you have to remember 3 or 4 important ones. One is what we call agrimycin, streptomycin, tetracycline, cyclohexamide. So these are some of the bactericides that have been under use. But the thing is, bactericides are very effective in controlling the spread of the disease. But the thing is, if you use them indiscriminately, there is a very possibility for the disease resistance to develop. So hence, they have to be used under the strict supervision of agricultural scientists. Then third group is what we call nematicides. Nematicides that control the population of nematodes. So nematodes are, they are generally present inside the soil, and they cause many tumors and also galls, and they attack, root systems. Especially we have seen in case of, say for example, tobacco, etc. These nematicides are found to be causing a lot of damage. So the chemicals that are used to control nematodes are called nematicides. And these nematicides, generally they are used as what we call as a soil treatment, and also they are used as soil fumigation. So by using soil for mixing with the soil and also for using as soil fumigants, you can check. It has been proved that they check not only the spread of the nematicides, but also they are found to be effective in controlling even various pests and pathogens. That is fungal pathogens. So you have to remember 2 or 3 important nematicides. One is methyl bromide is there, chloropicrin. So chloropicrin and methyl bromide are very effective, and these carbamates are very effective in controlling these nematicides. Then the other important group, the fourth group is what we call insecticides. As the name indicates, these are effective in controlling various insects. So insects could be of many: grasshoppers, stem borers, root borers, fruit borers, thrips, jacids, whiteflies, whatnot. So many types of these insects are there that are found to cause a lot of damage, not only to the standing crops, but also in the storage areas, in the godowns. So now insecticides are primarily classified into, based on their chemistry, some are organochlorides. The other is what we call organophosphates. And the third one is carbamates. These are 3 primary groups based on their chemistry. The important insecticides are, one is organochlorides, organophosphates, and carbamates. So the organochlorides, good example for organochlorides is the DDT, dichlorodiphenyl trichloroethane. This is an chloride. Of course, This has been proved to be very effective in controlling pests, but it has a disastrous effect on environment. So hence, DDT has been banned in so many Western countries, but still in India and other third world countries, it is being in use because the DDT has what we call biomagnification and found to be very dangerous for various animals and also human beings. DDT1. Then organophosphates, that is a phosphate containing insecticides. Dichlorovas, chlorpyrifos, monocrotophos. Phos means phosphate containing insecticide. And carbamates in carbofuran. Carbofuran is an example for carbonyl, endosulfan, dichlorovas, chloros, and methyl dimetone, dimethoate, methyl parathion. These are some of the uh, phosphate containing and carbamate containing insecticides. Okay, so these uh, insecticides, pesticides, and what we call bactericides, and fungicides, nematicides. These are available in various formulations. They can be used either in the form of dust for the seed treatment or for the soil treatment or in the form of, spraying. Generally, they are available in the form of what we call emulsification, emulsifiers. So that active principle, you mix it in water and make a solution by using sprayers, you spray on your field. So while spraying also, we have to follow whatever the direction that is given on the pocket or on the vial. So the concentration that has to be there. And even while spraying also, the farmers are advised to cover their nose. And generally, they are not to be sprayed against the wind. Towards the wind side, it has to be there. Or else, there is every possibility for the inhalation of these insecticide, and it may cause, it may found to be fatal, or the sprayer or the farmer who sprays, may lose consciousness sometimes. So one should be very, very cautious in making these, concentrations and spraying, etc. One has to be very, very careful. So this is chemical. Of course, the chemical fertilizers, coming to the positive and the negative side, the chemical fertilizers, they're quite effective. They're quite effective, and you can get the results instantly. Within a week time, you can get results. You can fastly, you can control, you can protect your crop. But the thing is, nowadays they are found to be very, very costly. It's a costly affair, and one has to have some knowledge making various concentrations while spraying, etc. One should be there. And when you spray them, sometimes, or nowadays, many times, the chemical may kill not only your pathogen, but also beneficial organisms. Now you might have read, papers and other print media, electronic media, some important bees populations, bees have been eliminated. Bee population, sparrows are eliminated. So the food chain gets disturbed. Bee population lost means because bees are found to be very, very beneficial for the farmers because they are the pollinators. They help in cross-pollination. If bee population is lost, if butterfly population is lost, sparrow population is lost, means your crop production may get altered in the coming years. So while using these pesticides, insecticides, bactericides, one should be very, very careful. Though they are effective, but they have, disastrous effect on environment. That you should keep in mind. Then we move to the ecological, that is ecological pest management practice. So ecological means, so this the environmental friendly. The chemical control is not environmental friendly. I told you it is disastrous for the environment. But just opposite to that is biological control. I told you curative methods are of two important principal types. One is chemical method, the other is biological method. This biological method is what we call an environmentally sound and effective method in reducing or mitigating the pests and pest effects. And they are quite effective in controlling the enemies and protecting our beneficial organisms. So this International Organization of Biological Control, there is an organization that defines this biological control as use of living organisms or their products to prevent or reduce the losses caused by harmful organisms. So here, what do you mean by that? Use of biological organisms or their byproducts, using them in controlling the various pests and various pathogens that are inimical to our crops. This is an environmental sorry, this is an environmental practice, environmental friendly practice, use of biological. So in nature, you see, if you look at nature, we have a lot number of species. If you observe very closely, so in nature, we have food chains and food webs. Food chains and food webs. Say, for example, if you look at if you go to a field, if your field is infested with, say, for example, grasshoppers or leaf hoppers, they will damage. But to control their population, what we have, uh, fly catchers. What are the fly catchers? Generally, your frogs and birds. If the bird population increases, or if frog population, rat population increases, that is also not good. Then to control their population, what we have in nature, snakes are there. Snakes feed on these rats and frogs, etc. They live on them. And to control the snake population, what we have? We have eagles or monks. They control their population. So in nature, there is what we call checks and balances operating. Nature always, checks the various populations by checks and balances. Always there is a checks and balances are there. So if one population increases, outnumbers the other population, it leads to what we call disastrous. It becomes a disastrous nature. So here, this biological control is preferred when compared to chemical control. So here, in biological control, what we do? You encourage the population of natural enemies instead of going for what we call these synthetic pesticides, synthetic bactericides, synthetic insecticides. You go for what we call natural enemies or natural products. So the natural products or natural enemies are they are parasites. They could be predators. They could be pathogens. They could be competitors. There are 4 groups of biological control is done by these 4 groups of organisms. One is parasites, predators, then pathogens, and the last one is the competitors. Parasites means that, see, for example, we have what we call bacteria. Bacteria causes so many diseases. But even bacteria are parasitized by, or bacteria are killed by viruses. What we call bactericides or bacteriovirus viruses. So the virus attacks a bacterium and makes an entry and uses all its machinery, absorption, penetration, assemblage and lysis, reproduction or multiplication, viruses. So each organism is controlled by another organism in nature. Hence, I told you checks and balances are there. So we have certain parasites. These are parasitic on disease-causing organisms. We have, say, for example, we have bacteria. So there are other bacteria which are parasitic, which control the other bacteria. We have fungal pathogens, and we have certain fungal species that grow or that check the proliferation or reproduction of other fungi. Okay, so we have what we call, predators. Predators are, see, for example, birds, dragonflies, beetles, webs, toads. So these are all, they pick. See, birds are there means birds fly, they pick the hoppers, aphids from the surface. They catch them, they eat them. Thus, they reduce the population of these pests. So here, and the last one is competitors. So disease causing, all are not disease causing organism, but some are. Say, for example, if you take human gut, we have hundreds of species of bacteria. So most of the bacteria are beneficial to us, but very few are inimical to us. So when the population of always beneficials are outnumber the pathogens. If the pathogen number increases, we suffer from a disease. So here what I mean to say is, in nature, there are so many natural enemies operating. They kill, they check the spread of disease-causing organisms. So here our intention is to increase the population of these natural enemies in the environment. So thereby, you can check them. You can check their population. So this is how we can check the population of various disease-causing organisms. So they are what we call predators. Predators means, for example, dragonflies, beetles, spiders, mites, monkeys, toads, birds like crow, sparrow. What they do? They pick disease-causing organisms, that is your pests, the young ones, the caterpillars. They spot them, they eat them. Thus they will reduce. So here our intention is to increase the population of these predators, parasites, and pathogens, and the competitors in our environment. Thereby, by using them, you can check the spread of the disease, and you can keep their population under economic threshold. So here, the biological control, we have three important strategies. One is what we call importation, augmentation, and conservation. You have to understand the meaning of these three. Importation. See, for example, we have nowadays, because of better transportation, etc., because of the world has become a village like. You can have your tea here, you can have your breakfast in one country, you can go for your lunch in another country, you can go for your dinner to far London. So within a day, you can travel hundreds and thousands of miles. So here, the importation means, here what we mean is, we have so many what we call exotic species that are not indigenous. See, for example, we have so many. For example, what we call Parthenium, Congress grass, what we call. Parthenium hysterophorus, a member of Asteraceae. It doesn't belong to India. And of course, this Opensia delineae, that is there. And Eichhornia crassipus, Lantana camera. These are all indigenous. They came from different countries, from America, from Australia and other countries. They came here and they got established here. And they have, because of fast, reproductive ability, they are occupying larger areas and posing a great threat to our indigenous plants, crop plants, and other wild plants. So here, importation is you import the natural enemies from other countries. For example, we imported beetles that eat the leaves of Parthenium, that eat the Opensia, that eat the leaves of Eichhornia crassipus. So we don't have, because they do not belong to our country. They are exotic. So if you go to the original country, there you will find their enemies. So here our intention is to import them, to encourage their population in our area. This is what we call importation. Augmentation means you maintain them and you release them into the environment. You preserve them in your greenhouses, etc., and you release them. Releasing will be of two times what we call inoculative release and inundative. Inoculative means phase-wise. Smaller doses you will be releasing. You will be checking the spread of these population, unwanted population. And inundative means releasing in large numbers, so that the disease causing or the unwanted plant population gets checked. This is what we call augmentation. And the third one is what we call conservation. Conservation is here. So when you import these foreign exotic ones, of course, they are able to check. But even in our area, in due course, even we have our local enemies. So our aim is to preserve them. To preserve them in our environment. So generally in research institutes and agricultural areas, their population, the various predators, these are dragonflies or beetles, webs, or other predator population will be maintained. So a small crop is there. And there, these populations are released. Natural enemies are released. And their population is kept. So as and when they are needed, they'll be taken and they'll be released in the area where their services are needed. So now these types of biocontrol, generally we have what we call predators. I told you, predators are, they feed directly on the insects. They spot them, they carry them, they remove them and eat them. Thereby they check their population. And generally I told you, beetles, spiders, mites, monkeys, frogs, and crows and sparrows fall under this. Then parasitoids. So parasitoids means these are the fungal members. These are the fungal members that parasitize on the insects. So the insect population is checked by the predators, but not completely they'll be able to remove them. But to control their population, what we do, we release certain fungal members that grow on the insects, on their body and also inside their body. Thereby, they cause a lot of damage to them. So the infected caterpillar or young ones, they become sluggish. They lose their color. Their digesting ability reduces. Ultimately, they get paralyzed and get killed. So this is the release of certain, pathogens. Especially, it could be what we call a fungus. For example, trichogamma is there, and bacteria, certain bacteria are there. Certain viruses such as verticillium is there. Then Bavaria bassiana is there. Metarhizium. These are the fungal members that are released in the environment, and they are made to grow. They are released in the form of spores. They germinate on the body of the caterpillar, and they make an entry into the gut and other areas, and their enzymatic activity checked. Digesting ability gets checked. So thereby they get paralyzed and get killed. And entomopathogens, certain bacteria, Bacillus thuringiensis, etc. We'll learn about this in tomorrow's class, wherein Bacillus thuringiensis has the ability to produce certain endotoxins. So the genes of this Bacillus are, uh, transferred into our croplands, and when a pest eats that, and they eat whenever it eats, and along that, it also gets ingested the inactive proteins after entering into their gut. They become active. These inactive proteins become active in the gut environment, and thereby they cause damage to its gut, and ultimately the insect dies. So these are all entomopathogenic. And we have also what we call baculoviruses. Even viruses are there. For example, nucleopolyhedral viruses are there. These viruses are released into the area. They enter into the body of these pests and also bacteria, and thereby they check the population of bacteria and also the pests. So entomofungi is there. I told you already. So pathogenic nematodes are also there. Even certain nematodes infecting bacteria, nematode infecting fungi are there. They are released into that area, and they contaminate. They make an entry. They grow either on the body of the nematode, or they make an entry into the gut area of the nematode, and there they secrete the materials, active principle, which are inimical to the digestive system and also respiratory system, etc. Thereby, the insect gets, or the nematode gets paralyzed and get killed. So we have what we call plant pathogens also. We have certain pathogens, certain certain viruses, certain bacteria. They compete with the other bacteria. So normal bacteria and inimical bacteria. So when disease-causing organisms are there into that area, we release certain disease-causing organisms. So both of them will compete. So thereby you can reduce their effect on the